Millions and millions of deaths have been brought upon by the coronavirus, especially during the year of 2020. And for months on end, scientists have been working extremely hard in order to create a vaccine to save us from this deadly virus. By the grace of God, they have finally been able to do so, but how did they do it? Could the recombinant DNA technology have been the key to their success? My name is Beatrice Nikole Gumahin, and I'm here to talk about how the recombinant DNA technology can be helpful in creating a vaccine against COVID-19. Based on a study conducted by Khan et al., the recombinant DNA technology comprises genetic material outside an organism to obtain enhanced and desirable characteristics in living organisms or as their products. Now, this works through the insertion of the DNA fragments from a variety of sources, having a desirable gene sequence through appropriate vector. Usually, it would take years to create a vaccine, but when scientists switched to genetic engineering, it only took months for them to create a vaccine against the coronavirus. There are two types of vaccines that use genetic engineering. First is the mRNA or the BioNTech, Pfizer, and Moderna vaccines. Now, since the messenger RNA just carries instructions for the assembly of proteins from the DNA template to the ribosomes, it is useful for vaccines because then scientists can easily reconstruct specific genetic sequences that encode for the proteins that are unique to the invading virus. Now, in the case of COVID, this is the familiar spike protein that enables the virus to enter the human cells. But how does this work? Well, first, the mRNA vaccines prompts a few of your cells near the injection site to produce the spike protein. This will then prime your immune system to create antibodies and T cells that will fight off the real coronavirus when it comes. This approach trains your immune system directly with a single protein. The next one is the adenovirus or the Oxford vaccine, which uses a viral vector approach. The scientists took an adenovirus, which is a type of pathogen that causes a common cold, and spliced it in the same spike protein genetic sequence from the coronavirus. The adenovirus simply serves as a vehicle to get the genetic sequence into your cells. Now, this is where the genetic engineering comes along. First, the vector viruses are stripped off of any genes that might harm you or cause replication. Then, using the recombinant DNA, the coronavirus spike protein genes are added. With that being said, I personally believe that through recombinant DNA technology, we have been able to produce such strong vaccines in a short amount of time. Hence why I believe that it is helpful. That concludes my presentation. Thank you.